my pleasure to introduce our next speaker. He is my colleague, Raymond Wong. Dr. Wong got his PhD from USC in immunology. He then went on into industry to work for Mankind for a little while. And 2 years ago, he joined the Pacific Meso Center. And today he's going to be talking to us about mesenchymal stem cells.
Right. Do I need this on or? Okay. Okay. Got it. Okay. Well, good afternoon, everyone. Hope everyone isn't suffering from too much food coma after that good lunch. So I'm going to be discussing some early exploratory work on this idea of engineering mesenchymal stem cells to deliver immunotherapeutic treatments that are targeted towards the mesothelioma stroma. So when we hear the term stem cell therapy, quite often the first thing that comes to mind is regenerative medicine, such as treating blood disorders, repairing tissues and organs, such as spray-on skin to treat severe burns, growing corneas in the lab for eye transplants, repairing heart muscle, regenerating liver tissue, restoring bone and cartilage and joints, treating spinal cord injuries. Maybe we think of this guy, Peyton Manning, whose career was all but left for dead in 2011 due to a neck injury. So he flies over to Germany, receives a so-called stem cell therapy, in which presumably stem cells were harvested from his body fat and reinfused back into him to treat his neck injury. And then he comes back in 2013, a couple years later, and just completely obliterates all the major single-season NFL passing records. Now, we don't know if his unconventional treatment had anything to do with his remarkable recovery. For all we know, that he might have just had a really good orthopedic surgeon and a really good physical therapist. But nonetheless, when we hear the term stem cell therapy, typically we think about regenerative medicine. But there are also other uses of stem cells to treat human disease, in particular certain inflammatory conditions such as arthritis, inflammatory bowel disease, graft versus host disease, which can sometimes arise from tissue and organ transplants. But first, let's clarify the major types of stem cells and where they can be harvested from. First, there are embryonic stem cells. Of course, working with embryos is fraught with very major moral and ethical concerns. We don't work with embryos in our lab. Then there are adult tissue-derived stem cells, which can be harvested from developed tissues such as bone marrow, body fat, umbilical cord blood, and term placentas. Now, we work with a type of adult tissue-derived stem cell called mesenchymal stem cells, which can be harvested from all four of these tissues that I mentioned. We work with mesenchymal stem cells that can be harvested specifically from full-term placentas. So placenta is part of the afterbirth, after delivery of a newborn, that's typically discarded as medical waste, but is very rich in mesenchymal stem cells. Now, we work with term placentas because they're easier to obtain as opposed to bone marrow, body fat, and it provides more raw tissue to work with than umbilical cord blood. So mesenchymal stem cells are already being used clinically to treat certain serious inflammatory conditions, most notably graft versus host disease in children, which can arise from tissue or organ transplants. And this is a disease in which the immune cells of the donor can sometimes begin attacking the tissues of the recipient. Now, there is one treatment for graft versus host disease involving the use of third-party mesenchymal stem cells harvested from the bone marrow of healthy donors, meaning the general public. They're manufactured to clinical grade standard and then infused into children suffering from graft versus host disease. Now, this is actually a commercialized therapy that's available for sale in at least two countries that I know of, Canada and New Zealand. Not yet in the US, but certainly there's precedent for their safe and effective use to treat human disease. Not yet in a cancer setting, but certainly in regenerative medicine and the inflammation fields. So what are mesenchymal stem cells, or just MSCs? Well, these are adult tissue-derived stem cells that are multipotent, meaning that they can develop into different cell types. At minimum, they can develop into three types of cells: fat, cartilage, and bone, depending on the particular harvesting methods and the culturing protocols, they might even develop into muscles such as heart muscle, and potentially even neurons and blood vessels. So one of the natural functions of MSCs in the body is to serve as a reservoir of cells to maintain and regenerate tissue. And again, MSCs can be harvested from a number of different tissues, most often bone marrow, but also body fat, umbilical cord blood, and what we work with, term placenta. So some important properties of MSCs that make them potentially useful for treating cancer. First and foremost, they have a natural attraction for damaged or otherwise abnormal tissue, and that's consistent with their primary function in tissue maintenance and regeneration. In animal tumor models, MSCs can engraft in tumors and stably integrate into the tumor stroma, and because of that observation, there's been an increasing amount of preclinical research and also discussion on potentially using MSCs as a drug delivery vehicle to improve the targeting and potency of anti-tumor compounds. And this, in theory, would also reduce drug-related side effects, considering that the treatment would be specifically or preferentially targeted towards the tumor. Now, what I just said, it sounds like a really attractive and provocative concept, but right now it's just a theory based on preclinical observations in animals. There are still many unknowns regarding the basic biology of human MSCs and their potential utility to safely and effectively treat cancer. But we do know in animal tumor models that MSCs engineered to deliver some type of therapeutic compound. It's usually a protein. When these engineered MSCs are injected into tumor-bearing mice, either through the tail vein to simulate an IV infusion or injected directly into the tumor, these engineered MSCs can stably engraft in transplanted tumors, release a therapeutic payload in the tumor, and in a wide variety of tumor models, this can result in delay of tumor growth or even shrinkage of established tumors. Now, currently, we don't know what would be the most optimal route of administration in humans would be. In the case of pleural meso, intuitively, we think that the safest and most effective route would be to directly instill therapeutic MSCs into the chest cavity, especially right after surgery when all visible tumors are removed, leaving behind only the surgical margins to treat. So there's still a lot of basic questions regarding the utility of MSCs and where to actually get them to manufacture a potential clinical therapy to treat cancer. Now, as far as the source of MSCs, ideally, the patient's own MSCs would be the most ideal. Since it comes from their own body, there aren't any tissue compatibility issues. The challenge with harvesting MSCs from cancer patients is that the quality or the quantity of MSCs that we can harvest from their bodies might be suboptimal due to the patient's age or any prior therapy such as radiation or chemo, which might negatively affect the quality or quantity of MSCs that we can harvest from these patients. So in practicality, this leaves us with using third-party MSCs harvested from healthy donors. And there's already been numerous clinical trials using third-party MSCs in the regenerative medicine and the inflammation fields. And overall, the data coming out of these clinical trials suggests that third-party MSCs are generally well tolerated with no major safety issues that have been identified. Why are they well tolerated in genetically unrelated recipients and not rejected? Most likely because MSCs are generally what we call immune evasive and not easily recognized as foreign tissue in the body. Now, there's still a lot of questions regarding the utility of MSCs for delivering therapeutic proteins towards tumors. So right now, there's still a lot of basic questions that we need to answer regarding what are the most optimal ways to harvest and engineer MSCs to deliver therapeutic proteins in order to be able to predict their potential safety and efficacy for treating cancer. So as I mentioned, MSCs can come from a variety of different tissue sources. We work with full-term placentas because they're easier to obtain than other tissue types such as bone marrow, body fat, and they provide more raw tissue to work with. than, say, umbilical cord blood. So harvesting MSCs from placentas is a fairly straightforward process. We digest the entire organ, place the entire cell mixture into cell culture flask, add in specialized MSC growth medium, and within 2 to 3 weeks, passaging and clearing out unwanted cells, we're left with an expanded population of highly purified MSCs, which can be frozen away and rethawed for use. So as far as MSC quality control, there are consensus minimal criteria and guidelines that we follow to ensure that we have indeed bona fide MSCs. First, they need to be adherent in culture, meaning that they need to stick to plastic cell culture flask. And this actually facilitates the isolation process because all the unwanted cells are typically loose and floating. They can be passively washed off. They need to be self-renewing or have the ability to replicate. That's a given since that's one of the fundamental properties of stem cells. They need to express at least 3 MSC-associated markers, which can be measured by fluorescent labeling. They need to be negative for blood cell markers, and they need to be able to differentiate or develop into these 3 cell types: fat, cartilage, and bone. So using the placental processing protocol that we've adapted for use in our lab, we've been able to isolate highly purified MSCs that express all the correct surface markers. They don't express any markers that they're not supposed to express. And we get a lot of MSCs from a single placenta. Theoretically, a single placenta provides enough cells to treat multiple patients. For identification purposes, we've also confirmed that these MSCs that we harvest from placentas can differentiate or develop into fat cells, bone cells, and cartilage. And again, this is purely for identification purposes. This particular quality control process will undoubtedly be part of any clinical grade manufacturing process for creating a clinical therapy potentially to be used to treat meso. So currently, we have a frozen repository of placental-derived MSCs, and currently we're working on our engineering protocols to induce the MSCs into producing therapeutic proteins. And again, this is based on the theory that MSCs have a natural attraction for damaged or otherwise abnormal tissue, including tumors. And in theory, this allows them to potentially be used as drug delivery vehicles to improve the targeting and potency of known anti-tumor compounds. As far as candidate therapeutics that MSCs can potentially deliver to tumors such as meso, where there are pro-drug converting enzymes, which some other groups are looking into, we're more interested in proteins that are directly therapeutic against tumors, or those that can activate a patient's immune system to recognize and mount an attack on their tumors. Basically a form of immunotherapy, which has undoubtedly been one of the more important breakthroughs in clinical cancer research in recent years. One of the more promising immunotherapeutic proteins that's relevant for treating meso is interferon alpha. As far as the anti-tumor effects of interferon alpha, it's directly cytotoxic or cytostatic against certain tumors, meaning that it can directly kill tumors or inhibit their growth. It can impair tumor blood supply, it can activate the immune system, and it's already a commercialized drug and FDA approved for the treatment of melanoma, kidney cancer, and certain blood cancers. As far as the effectiveness of interferon alpha in treating pleural meso, it appears that direct instillation into the chest cavity thus far seems to be the most effective route of administration. And Dr. Dan Sturman at the University of Pennsylvania has already conducted small pilot clinical trials and still is conducting clinical trials to my knowledge, where he's utilized an adenovirus carrying the human interferon alpha gene, and he directly instills it into the chest cavity of pleural meso patients. And even though his trials are small, there's some encouraging clinical evidence of tumor control, in some cases, even shrinkage of established lesions. The problem with using adenovirus to deliver interferon alpha is that it's a virus. The patient's immune systems typically generate neutralizing antibodies against the virus after the first cycle of treatment, such that subsequent repeat treatments with the adenovirus are rendered ineffective. So treating with adenovirus is typically a one-shot deal, but MSCs, on the other hand, as I mentioned earlier, can evade the immune system to a certain extent, and they're generally well tolerated even in genetically unrelated recipients. So MSCs might serve as a potentially improved vehicle for delivering interferon alpha towards tumors, and especially if it turns out, or especially if it holds true that MSCs can stably engraft and migrate deep into the tumor stroma. So this is where we are right now in this project. We're currently optimizing our engineering protocol to induce the MSCs into producing interferon alpha, and coincidentally, we also use adenovirus carrying the human interferon alpha gene. We infect our MSCs with the virus, wash off excess virus, and then our MSCs serve as a vehicle for production of interferon alpha. And the end goal is to measure how these engineered MSCs influence the growth and overall biology of human mesothelioma cells grown in 3D and embedded in a gel-like matrix that simulates invasion into surrounding tissue. But first, there's still some engineering optimizations that we need to work on for the purposes of inducing our MSCs to produce maximal interferon alpha. Ideally, we want to induce 100% of our MSCs into producing interferon alpha, and we can certainly achieve that for the jellyfish derived green fluorescence protein, which we use as a positive control. So these MSCs are definitely competent and capable of efficiently producing an ectopic protein. As far as producing interferon alpha, we've only been able to achieve up to about 40% production efficiency, meaning about 40% of the MSCs are capable of producing interferon alpha. So still some room for improvement. We want to hit 100% since they're capable of that level of production efficiency. So this adenoviral engineering optimization of the MSCs, this phase of the project is definitely is currently in progress. So as far as measuring potency of engineered MSCs against meso, our strategy is to utilize human mesothelioma cells grown in 3D. And this will hopefully allow us to simulate tumor biology as it exists in the body. And this allows us also to use a completely human tissue experimental system to hopefully mimic as close as possible the interactions between mesothelioma cells and also the therapeutic MSCs. Now, these 3D tumor cell cultures are microscale in size. You see the scale bar here. That's 0.5 millimeter. So quite literally, we can plate out hundreds, perhaps even thousands of these 3D tumor-like cultures simultaneously at a time, and that allows us to be able to test a wide breadth of therapeutic conditions with the purposes of obtaining as much data as possible so that we can understand how these therapeutic MSCs might influence the overall biology of mesothelioma as it appears in the body. So how do we measure potency of engineered MSCs against meso using this 3D mesothelioma model? Well, we can measure alterations in growth, objective growth or size or invasive activity of these 3D tumor-like cultures or 3D tumor cell cultures. That's one level of measuring potency of engineered MSCs. We can also look at how MSC-based therapies influence the molecular signature of mesothelioma cells grown in 3D. And by molecular signature, what I'm referring to is how genes are regulated in response to an experimental treatment. And this particular information could potentially help us predict how tumors in the body, such as meso, might actually respond to this particular therapy. So what's of most interest us in meso is the immunological signature of the tumors and how that might be altered by MSC-based therapies that deliver immunotherapeutic treatments such as interferon alpha. So we and also other groups have found that when we culture mesothelioma cells in 3D, that their baseline immunological signature appears closer to what is actually found in clinical tumors in the body compared to more traditional two-dimensional culture methods. So in a nutshell, we believe that by culturing mesothelioma cells in 3D, this offers a more accurate physiological simulation of mesothelioma tumors in the body, and hopefully this will help us more accurately predict how patients or how in vivo tumors might actually respond to this particular therapy. So to summarize, the most important take-home message is that the placenta, which is part of the afterbirth and normally discarded as medical waste, it's a rich source. It provides a rich source of mesenchymal stem cells, which can potentially be used as a vehicle to deliver therapeutics that are targeted towards the mesothelioma stroma. We're starting with interferon alpha since it's already being evaluated for treating meso. It's already a commercialized drug, so there's a lot that's known about it already as far as safety and efficacy, but we're certainly open to looking at other potential immunotherapeutics that we could deliver using MSCs. We do, as far as testing potency of engineered MSCs, we do have a model in which mesothelioma cells and also other tumor cell lines are grown in 3D. And we believe that this particular approach is an improved model to predict response to therapies. One of our other goals in parallel is to build more complex 3D models of meso that include other cellular components found in clinical tumors, including connective tissue, blood vessels, and immune cells. So this in and of itself, building complex 3D models for meso in and of itself is a major undertaking. But we can take our cue. But the good news is that other groups have also built such complex models. So we can certainly take our cues from those groups as far as building more physiologically relevant 3D models for meso. So as far as the long-term outlook of the Pacific Meso Center's role in MSc research for mesothelioma, we're a small group, so certainly we can develop the initial preclinical proof of concept on a small scale, focusing on the niche of using placental MSCs to deliver immunotherapeutics. Eventually, we'll disseminate that information through peer-reviewed publications, and hopefully it'll be picked up by entities that have the resources to potentially move it into clinical trials if there's enough interest there. So it'll certainly take a community to actually translate this particular theory from a theory or a concept into an actual real treatment. But that's still years down the road. There's still a lot of basic research that still needs to be done. So lastly, the